Good afternoon and welcome to our webinar entitled Early Drug Development Programs, the Need for Translational Medicine and Proven Regulatory Strategies. My name is Janine Estrada and I will be your moderator today. Today's webinar is being presented by Clinilabs Drug Development Corporation. Before I introduce today's speaker, I would like to cover a few housekeeping items. Today's webinar is being recorded and we will be able to share a link with you after the event is complete. We welcome you to revisit the content yourself and share it with colleagues. We also invite your comments and questions. Please identify the interaction panel on the right of your webinar screen, and if you think of a question at any point during the presentation, please add it there and we will hold it for our Q&A portion at the end of the event. Without further ado, I would like to introduce our presenter, Dr. Luana Pesco-Kopelitz. Dr. Pesco-Kopelitz is Chief Medical and Scientific Officer of Duck Flats Pharma in Flemington, New Jersey, which specializes in adult and pediatric translational medicine, clinical pharmacokinetics, and non-clinical development in all therapeutic areas, including oncology. Dr. Pesco-Kopelitz has received numerous awards in clinical research throughout her career. She has served on the FDA, as well as the Pharmaceutical Research and Manufacturers of America QT Working Group Committee. Since 2007, she has been a member of the Advisory Committee of the FDA Cardiac Safety Research Consortium. Since 2015, she is also an invited member of the International Neonatal Consortium of the Critical Path Institute for FDA, EMA, PMDA. Dr. Pesco-Kopelitz, please take it away.
Good morning, and I appreciate this opportunity to present this talk about early drug development programs and the need for translational medicine and its effect on regulatory strategies. This slide just shows my background in working with multiple pharmaceutical and biotechnology companies, and my field is actually translational medicine, clinical pharmacology, also non-clinical, and I certainly worked in regulatory strategy. So the objective of this talk is to understand the role of translational medicine in an overall drug development program, the preparation of a successful IND and IND package, and also preparation for a successful pre-IND meeting with the FDA. As many of you may know, a new chemical entity or biotechnology compound almost always includes translational medicine as part of the strategy. I think many times people will think that a drug development program, they'll focus on the pivotal phase 3 safety and efficacy studies, but there are many other supportive areas that have to support the phase 3 program, and that include the chemistry, manufacturing controls, CMC, as well as non-clinical and early clinical. So for translational medicine, these areas include non-clinical pharmacology, toxicology, pharmacokinetics, including ADME studies, and the early clinical research, the phase 1 and early phase 2 research studies in either patients or healthy volunteers. All of these parts of the development program that I've mentioned need to work together to address the preliminary efficacy and safety compound. This is also necessary when companies are developing older drugs for either new therapeutic applications or new patient populations. These are normally called 505(b)(2) applications. Key roles in translational medicine include, obviously, excellent research and development. The use of biomarkers and exploratory markers can also be very critical. Use of adaptive clinical design. And you may be using either healthy volunteers or patients, especially in the SAD, which is single ascending dose, or MAD, multiple ascending dose studies. So the actual drug designation and therapeutic area can impact the overall approval timeline and cost. So for example, for a new molecular entity or for certain biological compounds, you can often have the most protracted timeline because you have to develop everything de novo. When you're looking at a 505(b)(2), in general, those timelines can be shorter than a new molecular entity, but depending on the patient population and how the drug is going to be administered, you may not save as much time as you think. Now, an orphan drug application, which usually includes less than 200,000 patients, again, depending on how the studies are run, you may have a shorter timeline. Certainly with a breakthrough therapy where there is no known treatment, especially for a life-threatening disease, this could be a very short development timeline. First-time generics and even the second-line generics, of course, are usually just doing bioequivalent studies, and so they may have the shortest timeline. So just to give one an idea of what drugs have been approved by the Center for Drug Evaluation, in 2019, 48 novel drugs, meaning new chemical entities or novel biotechnology compounds, were approved. Of these, 20 were designated first in class, 21 for rare diseases or as orphan drugs. There are several regulatory pathways that can enhance the efficiency and expedite the development and approval of novel drugs. So of the aforementioned drugs, 17 were designated for fast track pathway, 13 drugs were breakthrough therapy, 28 drugs received priority review, and 9 drugs went through an accelerated review process. Now, there are certain limits that a company must meet in order to get these designations, and those are most likely discussed with the FDA a priori before the submissions come in. So this is just a snapshot of the annual novel drug approvals from 2010 to 2019. And as you can see, it does go up and down, but one can perceive perhaps a slight positive slope in the number of drugs approved. And it looks like 2018 was a significantly good year, but certainly an improvement, and the FDA is to be commended for being able to review and approve so many compounds, especially for novel drug submissions. So drugs entering clinical development most often fail to get approval due to some of the following reasons. First of all, as many as 57% of drugs just don't demonstrate efficacy once they get to the late phase 2, early phase 3 area. This has been a chronic problem with drug development in the past. Now, 20 years ago, that number was much higher. It was closer to 70, 80%. So it's come down to 57%, mostly because of improvements in the translational medicine arena. Some of the reasons, though, include flawed study design. They also include inappropriate statistical endpoints and underpowered clinical trials. Almost all of these particular points can be usually attended to quite well with the translational medicine program, as well as with very close interactions with the FDA, with their clinical groups, non-clinical groups, statisticians. Safety concerns is only 17%, meaning that there is a safety signal that wasn't really noted prior to the phase 3 program. And then it becomes noted when you get to phase 3. And sometimes they're only apparent in those larger phase 3 clinical trials. But bear in mind, it's only 17%. In my experience, I've seen that if you look very closely at the results of the translational medicine program, there may be a hint in there that someone may have overlooked. It's quite rare that I've seen where you have absolutely nothing in the translational medicine program and suddenly something large looms in phase 3. So it means that you also have to look more carefully. There are some drugs, and I've seen this also in oncology, where you don't have a flawed design. You don't have inappropriate statistical endpoints. You don't have any of that. It's just in phase 3, the drug doesn't work. You saw some efficacy signs in late phase 2, and it just didn't materialize. Were you lucky? Does a drug have effects, but only in a subpopulation of patients? Those are certainly possibilities. Therapeutic area also impacts drug approval. Oncology and CNS have lower approval rates compared to other drugs. And there are a number of reasons for that. They're much more complex trials, and endpoints are much more difficult to ascertain. So drug approval timelines per therapeutic area. There are substantial differences in approvals across therapeutic areas. And this just shows the average review time in months of the various drugs. As you can see, oncology is right there with one of the fastest review times. And often in oncology now, you are attending to diseases with life-threatening indications. So a particular oncology indication that is relapsed, meaning no prior treatment is helpful. If you have a drug that works in that area, that's considered life-threatening, and so you'll have less review times. So looking at specifically central nervous system or CNS drugs, the average time to develop is about 20% greater compared to other drugs in other therapeutic areas due to greater development challenges, many times incomplete understanding of the underlying pathophysiology of many CNS disorders. So you will know some of the pathways that are affected by a particular disease, but you don't know all of them. In many cases also, it is not easy to measure them, or there's some subjectivity in the measurement. So for example, in cardiovascular, if you measure blood pressure, you measure the blood pressure, you get two numbers, and it's quite evident what the endpoint is. But if you're looking at a visual analog scale for pain, where you have a lot of subjectivity in the assessment of that, that's going to cause a lot more variability. Except again, if it's a life-threatening or there's no prior treatment, in the CNS area, you're less likely to win fast-track approval because there are a lot of drugs that at least gives partial relief to a particular disease. And so you may not get fast-track approval. And because of the complexity of the studies and the fact that the phase 3 programs are very large number-wise, it can take much longer than, say, a drug in another therapeutic indication, say asthma or something else. So interestingly, as has been pointed out to me by colleagues, especially in big pharma, translational medicine actually could be the bottleneck between basic research and the phase 3 clinical program. And it also becomes the critical go, no-go decision for moving forward with a drug development program. However, it can also allow successful programs to accelerate, and we will call suboptimal programs to fail early. And failing early is a good thing in pharma. If you have a drug where you can determine by the end of early phase 2 that this is just not going to work, that saves billions of dollars in the long run. And those resources can be moved to other therapies that may have more benefit. Successful programs can impact some other areas such as the CMC, the chemistry, manufacturing, and controls. So there are different objectives. When many individuals will take a pill, for example, it's not exactly CNS, but people see advertisements on television with Nexium, the little purple pill. It took an incredible amount of money to develop that little purple pill and get it approved by the FDA. That takes an enormous amount of time and resources. If you develop, let's say, a very nice pill formulation and then get to phase 3 and find out the drug doesn't work, you've spent millions upon millions of dollars for nothing. At the same time, if you're not sure and you suddenly get fast track approval, and then the FDA wants to expedite your program, and now all you have is some powder being filled into capsules by hand with a spatula, that's not going to work either because you will never make enough drug for your program. So you need to balance these different objectives and at the same time not waste a lot of money. So in translational medicine, we start with the preclinical or non-clinical studies, and we start with animal studies, and then proposed testing in humans. You try to determine the first-in-human dose, which is a fairly complex process. Once FDA is comfortable with that, you're allowed to move forward with your phase 1 studies. Normally, you'll see safety, pharmacokinetics, if it's possible, biomarkers. I say initial efficacy. There are some diseases in which you will see changes in biomarkers in the patient population with the disease, but not in the healthy volunteers. But you may see it in healthy volunteers, it depends. And then you move on to your early phase 2, which is your proof of principle, your dose ranging and dose response. The phase 2 studies, the early phase 2 studies, that's one of the most critical areas in FDA, and they have made it known that this is one of the areas that pharma in its history has not done the best job of determining not just the fact that the drug works, the proof of principle, but the dose ranging and the dose response. So there also has to be medical and commercial viability. So it's not just the idea that, oh, well, we have a drug that works. Will the medical community accept it as a standard of care? Will doctors prescribe it? Will the patient think that the treatment is worthwhile? And there is a lot of research now on how patients perceive drug development and how they perceive their care. Patients are far more educated now with the internet and with the availability of medical literature on the internet. So you have a much more informed patient group, and their thoughts on this, their concerns need to be taken into account. Commercial viability. Can the sponsor sell the product? Does the development cost justify the price? And the biggest issue that I've seen is the issue with prescription drug plans, hospital formularies. Our group worked on a drug in an oncology indication that was actually approved as a fast track approval, and it improved the survivability from 12% to 65%. And these were patients that previously were quite ill, that were at the end of life, and now were out playing golf. So it was a huge boom. The oncology community, they were just thrilled. So interestingly, the company isn't making that much money off of it because none of the prescription plans will accept it because they say the cost is too high. So here's a drug that is life-saving, literally life-saving, giving extensive, good quality of life to patients, and yet only those patients, obviously the company will donate the drug to patients that need it. But many patients aren't aware of this and just forego and go with the older treatments that don't work. So the whole issue of prescription drug plans and hospital formularies has become a major issue. Potential development issues, difficult therapeutic areas. The two biggest, again, CNS and oncology. Again, the endpoints are there, but the mechanisms of action can be very varied. Oncology drugs have a fairly low success rate. It is probably has the most R&D worldwide as far as dollars than any other therapeutic area, but the success rate is somewhat low. The cost of discovery to potential drug approval is anywhere from $2 million to $3 billion, can take up to 12 years. This is just overall. In diseases in a small number of patients, you have orphan drugs. So those tend to be much more popular these days. These are specialized areas, a disease that's only found in 65 patients worldwide. Those drugs can get approved fairly easily, and a lot of companies start with that and then expand their indications after that's approved. Clinical trial failures. This is from a 2018 reference. Probability of success in moving from phase 1 to 2 is 66.4%. From moving from phase 2 to 3 is 58.3%. So there's a drop, but not a terrible drop. So it looks like we're doing a better job with the translational medicine piece than he did in the past. Just to be complete, I thought it was important to present the non-clinical components within translational medicine. All very critical. These are all cogs in the wheel, and all these areas support those two pivotal phase 3 efficacy and safety studies. So you need all of these. In general, there could be exceptions, of course, but in general, all of these. And if you're missing one piece, it can actually spell problems for your development program. Your bioanalytical methods, very, very critical. If you can measure the drug, compound, monoclonal antibody, whatever, in plasma, serum, whole blood, wherever you can find it, any biological specimen, that's very critical. That has to be validated. You have to conduct genotoxicity and reproductive toxicity. There are safety pharmacology, pharmacodynamic studies. These are mostly in the CNS, renal, cardiovascular. Toxicology. Toxicology is perhaps the most cookie cutter part of this system, but critical needs to be done. All of your pharmacokinetic, absorption, distribution, metabolism, excretion, transporter studies, there's in vitro, in vivo, that has to be done. Drug-drug interactions, even if you think, oh, it shouldn't interact, you need to do these studies. Clinical components, phase 1 studies normally are, again, for safety, usually pharmacokinetics, and then looking at biomarkers, surrogate endpoints, but probably safety and PK are the critical ones. And again, Phase 2 studies are, as I mentioned before, the proof of principle, efficacy, and dose ranging. The key component in these is predictability. What you ultimately want to see and what FDA wants to see is you need to see predictability from the non-clinical into the clinical. If it happens, sometimes there isn't. And if there isn't, but your drug does work, you just say there isn't. But you'd like to find a way where there's some link between those in any of the aforementioned areas. Your bioanalytical methods include your validation, your concentration ranges, endogenous compounds, biomarkers, diagnostic testing. All of these can be a piece of the bioanalytical methods portion. Genotoxicity, carcinogenicity, reproductive toxicity. I've just mentioned some points here on the slide. I think that these are well known to most people in the drug development area and are part of the toxicology program. Safety pharmacology and pharmacodynamics are also very critical. Also, I'd like to also mention, in addition to safety pharmacology, pharmacodynamics, it's important to also have some primary pharmacodynamic studies in animals to investigate the mode of action. Do they have the same type of disease? So, for example, a simpler example, diabetes. There are animal models for diabetes in which you can test a drug in them, and it may not be 100% predictive to what you'll see in diabetes in humans, let's say type 1 or type 2 diabetes, but it's close enough that you have an idea their drug has toxicity. It's also important in these pharmacodynamic studies to also get an idea of exposure, meaning pharmacokinetics, how much drug is in the blood. Is there a correlation between that concentration of drug in the blood and the changes that you see on the treatments for the pharmacodynamics? Toxicokinetics and pharmacokinetics. The only difference is that pharmacokinetics is generally concentrations of a particular drug at the therapeutic concentrations, and toxicokinetics is at much higher concentrations you'd see in a toxicology study. You certainly want to look at plasma protein binding, in vitro metabolic data, again, systemic exposure. Also for repeat dose as well as single dose, you need to characterize the human metabolites. And so therefore, you need to also characterize those non-clinically. If you see a human metabolite that you didn't see in animals, because man is the only individual that makes a metabolite, you need to study that metabolite in animal models. Normally, for toxicology, toxicokinetics, you do acute toxicity studies, you do single dose escalation, and you can limit it to the intended clinical route of administration unless there is an issue with severe toxicity or other things. The initial studies can be done as non-GLP studies, GLP meaning good laboratory practices. Then you have repeat dose studies. Those studies are usually in a rodent and a non-rodent species, usually rat and dog. There are quite a number of cases where those species cannot be used. Biologics, certain monoclonal antibodies often are studied in monkeys. There may be other reasons why other species can be used. These studies are usually performed as GLP. And in general, though, I must say that probably Big Pharma does both their acute and repeat dose studies as GLP anyway. Drug-drug interactions are becoming very, very critical, very important in the FDA. As a matter of fact, a guideline just came out literally yesterday by the FDA on looking at drug-drug interactions for monoclonal antibodies and other biological type compounds. And interestingly, there are effects. They're not direct, as you would see with the cytochrome P450 system with older drugs like ketoconazole or fluconazole or other drugs, but they do indeed exist. And so they're very important. Transporter studies, there is an FDA guideline on that. So again, these metabolic changes are very critical and they need to be known and understood in your program. If there are questions, it can literally hold up your entire program until that's known. So the first in human study is based on the calculation of a first dose. Nowadays, the SAD/MAD studies are included in your first study. It saves the client money and time. It saves an administrative cost. Sometimes a metabolism C14 study is considered the most prudent study to do in the first-in-man study. Sometimes a food interaction may be added to this first-in-man study. On a rare occasion, you might have a drug-drug interaction included in the study design, depending on, for example, if this is going to go in a patient population where they must be on a particular compound and there's a concern of safety, you may actually do your first-in-man with this drug on board in one of the periods of the study. Of course, you would generate your safety, pharmacokinetic, and/or pharmacodynamic data. Healthy volunteers or patients. If you have patients, preliminary efficacy can be studied. Can't say most oncology drugs. There are a lot of oncology drugs now that are safe enough to give in healthy volunteers, but many of them are given to cancer patients at the outset. Again, phase 2 studies are efficacy in the labeled patient population. They will be PK/PD, safety, efficacy, dose ranging, and dose response becomes a very, very critical part of the early phase 2 studies. Understanding your no effect dose and your maximum tolerated dose in the labeled population is very, very critical. And I have seen a number of programs over my career where this is not well defined, and FDA puts the phase 3 program on clinical hold, or even the late phase 2 program on clinical hold until the no effect dose is determined and the maximum tolerated dose. There are some drugs that don't have a no effect dose and don't have a maximum tolerated dose, or CMC becomes the rate limiter. That's okay as long as you've proven that. But they want to see very clear data. So pre-IND meeting is usually considered a type B meeting. It's to aim to see that the development program for your future clinical trials will be acceptable to the FDA. It's an excellent opportunity for sponsors to give feedback on their program from the FDA. It needs to be scheduled within 60 days FDA receipt of the written meeting request. And normally FDA will not grant more than one meeting type for each potential application. If there are issues, other things, you can get a type C meeting if it's deemed critical. So how's the meeting scheduled? The sponsor requests the meeting to the appropriate division. You give some limited information, background questions. They usually provide a formal response within 21 days. I've seen them go out to 21 days to respond. I've seen a response in a day and a half. It just depends on what they have on their schedule and their interest. They will, for all intents and purposes, the response will be, a meeting will be granted. However, a face-to-face meeting obviously now with COVID-19 is probably not possible, but they may be able to do Skype or something like that. Or you can have a teleconference. Often, written responses is all that you can have. Sometimes a meeting request is denied, not because they're trying to be difficult, it's because there's not enough information for them to be able to answer the questions that you're planning to ask them. They may see areas where they're deficient, and they'll just say the application that as you're going to present it is deficient, and so they will deny a request. But otherwise, if you have everything there, a meeting will be granted. The big issue a lot of companies have is they want that face-to-face meeting. It's good for the company as well to get that face-to-face feedback. And it includes a number of things you see here: the product name, an application number, structure, the type of meeting format, agenda, the list of individuals, and then suggested dates or times, but FDA will ultimately decide the date and the time. It used to be the meeting package was submitted 30 days prior to review. You'll see now it is now 45 days, which puts more pressure on the company to get everything in earlier. And again, the meeting package is usually I think it has to be within 200 pages, but you can also have appendices, which don't count as the meeting package. It's just to give the FDA enough information to review so they can discuss the package at the meeting. You need to include your CMC information, any additional clinical or non-clinical data, your investigator brochure, if you have any idea for pediatric study plans, and especially a gap analysis. So if you've in-licensed the compound, of what might be missing, what is needed. The investigator brochure, I'm sure is familiar to many, includes basically everything you know about that drug. It's not just the FDA that needs the investigator brochure. It is the Institutional Review Board or the Ethics Committee that will be tasked with reviewing the first-in-human protocol, as well as the investigator and other parties involved with the clinical trial. So close communication with the FDA early on really helps to reduce the chance that there might be a review hold or additional review. So once you submit your pre-IND package, the FDA has 30 days to respond. An IND is not approved. The FDA just allows you to move forward with your first-in-man study. So the day that it's sent in, you will then have 30 days. And some companies literally wait with bated breath. And as soon as they hear nothing on day 30, then they go to the investigator and say, okay, start enrolling the subjects or healthy volunteers. But there isn't a formal approval per se. If you hear nothing, you're good to go. FDA, if they really have a concern, they will contact you before the 30 days. So IND applications that are denied, meaning that they call and say, no, you can't move forward. You just don't have all your information in there. Your IB is missing. You have an inadequate protocol. You can give a small synopsis maybe for the pre-IND meeting, but most individuals have a full protocol. There are exceptions to that. Again, there always are in certain life-threatening indications, but if you do have a synopsis, it's about 30 pages of a synopsis. So you've just left out all of what I call the boilerplate of the ICH safety guidelines and that sort of thing, and they are assuming you will have that. The CMC areas can be deficient in some way where they say, no, you don't have enough stability data. We have some questions with your analytical methods. FDA may have said in their response to the pre-IND meeting, we want to see A, B, and C, and you don't provide that. They're concerned that your clinical studies might not follow all your GCP requirements. You say you in-license a drug from another country, and then they want to see that data, see those final reports, and you don't have all of that. If there's a concern with drug dependence or abuse potential, this is especially important with CNS drugs. They may want to be sure that that issue has been addressed up front and not wait till later in the program. That's missing. That could cause a clinical hold. That is my presentation, and thank you for the opportunity to present.
Thank you, Dr. Pesco-Koplitz. We have several questions that have been submitted by the audience. As a reminder, if you have a question that you have not yet submitted, please enter it in the questions panel and we will get through as many as time allows. Dr. Pesco-Koplitz, the first question is, are there differences in drug development programs amongst oncology compounds?
Actually, yes, there are. It will depend on the survival rate, the mortality rate, the difficulty of treatment of a particular cancer. So for example, for HER-positive breast cancer, you have women that have 20-year survival rates. So if you have a particular drug that would be useful in that particular indication, you will have to have probably a program almost as vigorous as one to treat hypertension or diabetes. However, if you have a medication that looks like it works very well in stage 4 ovarian cancer, then your development program will be much, much shorter. FDA will not require a lot of information. Because of this, a lot of first-in-human studies and development programs in oncology, you will now find are for drugs where the patients can no longer benefit from existing treatments, so they have no other recourse. They are considered, of course, life-threatening. So there's even an FDA guideline on what the toxicology program and the metabolism program for oncology compounds is somewhat truncated in a lot of the oncology development areas, and there's actually an FDA guideline, so you don't need every last thing that you would need for a drug, for example, or more chronic disease and chronic indication. So yes, indeed, depending on what you're treating, the type of cancer, its metastatic profile, does it metastasize easily or not? That will certainly depend on it. And truth be told, there's no cookie cutter, do this and you'll get a drug approved. Each drug development program is different. They are not all the same, even if it's in the same chemical class, same therapeutic class. There may be similarities, but they won't be exactly the same because each drug is unique.
The next question we have here is, does an oncology development program have the same requirements as other non-life-threatening indications?
Oh, so I probably answered that in my previous answer. So in general, when you have an oncology drug, again, if it's a more life-threatening indication, you do not have to do as extensive as a toxicology program compared to non-life-threatening indications. Also, the timing of when FDA would like to see certain data may be different. So, for example, certain data that's usually expected at the pre-IND stage, the FDA may be fine with receiving that in late phase 2 or early phase 3. So it's not just the types of studies, but it's also when when that data can be submitted. So indeed, yes, you often don't have to do as much work in the toxicology area in oncology compared to, say, a non-life-threatening or chronic indication.
Next question, can exemptions be made for the requirements of a drug development program in certain circumstances?
Yes, they can. I can speak about this because we were at the advisory committee meeting, so it's public. We were a major cog in the wheel for the approval of Narcan nasal spray. And so the FDA, well, first of all, naloxone is a well-known drug and approved, but it was obviously given in a new, a very different formulation intranasally. That development program was light years faster due to the opioid crisis that we presently have here in the United States. So they were much more accepting with less data, especially in the CMC area. I think we actually clinically, we had very solid data from the pharmacokinetics and that sort of thing. Again, if you have most life-threatening indications, the FDA will sit down with you. You don't want to obviously do any more harm, even in a life-threatening indication, but you look at things more quickly. And so there are certainly exceptions, but they have to be agreed to by the agency and worked through very carefully.
Next up, does the FDA accept all requests for Type A meetings? And if so, how are they assigned?
So a lot of it depends, well, certainly some of it depends on their scheduling, but in general, they will as long as you have everything there. So as long as you are not deficient in any of the pieces that they have, they will accept that. The issue will be if they accept the meeting, and you put that in quotes, your response may be a face-to-face, assuming there wasn't COVID-19, a teleconference, or just written responses only. Now, some clients will ask for written responses only, and the FDA is fine with that. But the face-to-face meeting is what many, many clients would like to have with the agency, and that's often very difficult to do just because there's so many hours in the day, and people have to travel and that sort of thing. Also costs the sponsor a lot more money, obviously, because you not only have the people from the company, you may have key investigators who are experts in the disease also coming with you and the like. So it's a combination, I guess, of different a combination of different scenarios. But in general, assuming your application is not deficient, they will try very hard to accommodate you.
Our next question is, how early do the pediatric requirements need to be considered in a drug development program?
So the pediatric requirements, which have been around now for some time, I actually have a talk just on that because I'm involved in a lot of pediatric research from neonates on up. You definitely have to have your pediatric program prior to going into phase 3. So by your end of phase 2 meeting, you must have that completed. And that's reviewed not just by the division, but by the pediatric division, which is a separate division of FDA. In some cases, depending on if there's a potential that this might be a very useful treatment in pediatrics, it is possible that FDA may ask for this information earlier than end of phase 2. So that is possible. Some companies now are being more proactive in addressing this earlier on, just so FDA knows that they're looking in that direction. We have a saying in the pediatric arena that the pediatric patients are the last true orphan drug group, except it's a very big group. They have many, many unmet needs, and the number of drugs that are labeled for pediatric use are very, very small. Now, part of that is because a lot of the diseases that are being treated, like prostate cancer, Alzheimer's disease, clearly are not pediatric drugs, but companies are moving in those directions, and so you don't have that. The other issue is that there are drugs that are used in the pediatric arena. It's already generic and no one's developing it because pediatric trials are difficult to do. So it's just not being looked at. But I would say by the book, before your end of phase 2 meeting, you need to have this worked out fairly well. And you can actually have a delay moving into phase 3 if this is not to the FDA's satisfaction. So it behooves one to consider this at least by the time you're moving into your early phase 2 program if you think pediatric patients may have a major benefit from your product.
And our last question of the day is, what major differences are seen with pediatric clinical trials versus clinical trials in adults?
Well, the first big thing, I guess, informed consent is probably a major area. Enrollment of patients, the protocols, there's obviously determining efficacy is going to be, if there's any subjectivity in determining efficacy, you're going to have a more difficult time with a 3-year-old than a 13-year-old. So most companies will often go down to age 12 for pediatric studies, and then have no labeling beyond that. A 12-year-old can usually give an accurate assessment of pain or discomfort or something like that. If it's something where it's purely physiological, so for example, you have some sort of congenital hypertensive state, that's something that you can measure in a neonate. Not that easily, but you can do that, but at least get some solid objective data. But it is hard often to treat certain pediatric diseases when you need some sort of subjective review. One of the big areas is the fact that you're trying to determine some sort of exposure response. So you obviously can't take 17 7 cc samples of blood from a 3-year-old. So one of the big hurdles that we've had to deal with is developing blood spot assays where you can have very, very tiny amounts of blood where you can actually measure drug concentrations. It's very time-consuming, very expensive, and it's a huge investment. So there are many different areas, but it's also very rewarding when you're able to develop a medication to treat children that have very serious diseases, and they're actually living longer or living without pain, or, you know, for the first time a child can go out and play with their parents because they feel well enough to do so. So the rewards are very great on the other side.
Thank you to everyone who joined us for today's webinar. Dr. Pesco-Koplitz, thank you so much for sharing your insight and expertise. If you have additional questions or would like to connect directly with the ClinLabs team, you can reach out to us via the contact information that we've provided, or you can reach out to us on LinkedIn, Facebook, or Instagram. Thank you so much for your time and have a wonderful rest of your day. Goodbye.